ladies and gentlemen. My name is Wilfried Schwab. I'm heading the group Biotechnology of Natural Products at the Technical University. The topic of my talk today is aroma glucoside production. You're probably wondering what aroma glucosides are. So we use this experiment, which consists of a small molecule, a volatile molecule, which is perceived by humans as flavor or fragrance, which is attached to a sugar molecule. The most simple glucose, but there can also be disaccharides or pentoses attached to this molecule. And our idea is to commercialize these compounds, and therefore I have to talk about these compounds. I cannot keep them secret, because we are trying to sell them. So these molecules are, these aroma glucosides are produced by the plant secondary metabolism. Plant secondary metabolism are compounds which are biosynthesized from primary metabolites, from carbohydrates, amino acids, and fatty acids derivatives. And there are three principal groups of secondary metabolites: terpenoids, the alkaloids, and the phenolics. Terpenoids, they are built basically by two building blocks which are generated by the mevalonate pathway or the desoxycellulose phosphate pathway. So terpenoids include the orange pigment beta-carotene. You probably know menthol, which is a cooling ingredient of food. And there is the anti-cancer drug Taxol. Vitamin A belongs to the terpenoids. You probably know about the new Coca-Cola, which contains steviosides. So the stevioside skeleton is a terpenoid, or stevioside contains a terpenoid structure. And there, of course, is cholesterol, which is also a terpenoid. Phenolics include compounds such as vanillin, the flavor molecule vanillin, salicylic acid, which is the basic skeleton of the drug aspirin. Then also the lignins have a phenolic structure. And there's also this red pigment, which you know from strawberry. This is an anthocyanin, and it also belongs to the phenolics. Alkaloids, all know the caffeine, which is an ingredient of the coffee. Then we have the toxins nicotine and also strychnine, and then we have the pain reliever morphine, and also a pigment or red pigments. In the case of the alkaloids, they are the beta-lains. So you see there is the secondary metabolism is very complex, but when you look at the biosynthetic pathways, there are three principal steps. And so the biochemistry is something like a combinatorial biochemistry. Due to these three different steps, a multitude of structures can be obtained. It's as simple as Lego biochemistry. So at the beginning, you have different building blocks. In the case of the terpenoids, you have basically only two building blocks. For the phenolics, you have some more building blocks, CoA esters, which can be derived from the Shikimi pathway, and the alkaloids are derived from building blocks, which are derivatives of amino acids. So when you connect these building blocks, you have a certain degree of freedom, depending on how you connect them to each other. So this is the first level of biodiversity. And then you can elongate the skeleton or the chain, and then you obtain a skeleton. And in the case of the terpenoids, prenyl transferases do this job and elongate the building blocks. Then there is a second level of biodiversity, the modification of the skeleton by oxidation, methylation, and glucosylation. So oxidation means that oxygen is inserted into the skeleton, into the structure, methylation, a CH3 group, and here glycosylation means that carbohydrates are attached to this basic skeleton. And at the end, although you are using the same basic building blocks, you can obtain different structure which show different biological functions. And in a similar way, also the aroma glucosides are obtained in the plant secondary metabolism. Let's have a closer look on terpenoids. Terpenoids are synthesized by plants, generally in two different departments, in the cytosol and also in plastids. And these basic building blocks, the DMADP and IDP, dimethyl allyl diphosphate and isopentenyl diphosphate, they are synthesized in the plastids by the desoxyxylulose phosphate pathway, and in the cytosol by the mevalonic pathway. And afterwards, these building blocks are connected to each other by the prenyl transferases, and so the chain becomes longer. The first products are the monoterpenoids. If three building blocks are attached to each other, then we have the sesquiterpenoids. They can be oxidized at the ER, the endoplasmatic reticulum, and obtain oxygen molecules. But these products are actually cytotoxic for the cell. In order to accumulate these compounds, nature has invented two different strategies. One strategy is to export these molecules into the outer space, into the extracellular space, and then glandular trichomes are formed. So they are on the leaf surface, and if you touch these glandulars, they become destroyed, and these volatile compounds, monoterpenoids and sesquiterpenoids, they evaporize, and then you there's a smell, or you perceive these volatiles as the smell of a plant. Another strategy is to glucosylate these compounds, attach a sugar molecule, make them more or better water soluble, and then they are stored in vacuoles. As you can see here, this red color shows you that the glucosides are stored in the plant vacuoles. And by these two strategies, a high concentration of terpenoids can be stored. And terpenoids isolated from plants, they have different structures, but the point for this slide is that most of them are glucosylated or glycosylated. Glucosylated means glucose is attached to this molecule. Glycosylated means a different sugar or even a disaccharide is attached to the molecule. So in case of the olefins, there of course is no chance to attach a carbohydrate. They occur only in the free form, but when alcohols are produced like geraniol, citronellol, linalool, or menthol, then you always will find the glucosylated form in plants. Also for the steviosides, they can carry different kinds of carbohydrates, and the sweetness and bitterness of these individual molecules, they differ. And so there are plans to biosynthesize only the derivative with the highest sweetness but the lowest bitterness. And here also the ginsenosides, which have some pharmaceutical properties, they also contain different carbohydrates, and the bioactivity of these molecules depends on the number, on the structure of the carbohydrates. You also consume a lot of terpene glucosides when you drink wine. Yeah? Grapes contain a number of aroma glucosides. They contain linalool attached to different carbohydrates, to glucose or 3 kinds of disaccharides, high level of geraniol, of free geraniol, and glucosylated geraniol, nerol, and citronellol. And this chart here shows that in some cases the plant cell accumulates 10-fold higher levels of the glucosides in comparison with the free alcohols. Here you see in blossom you have 200 milligram per kilogram tissue, whereas you have only about 10 milligram of the free alcohols. Yeah? Normally there are similar levels of the free and the glucosylated derivatives, yeah? But in some cases here also in the root the level of the glucoside exceeds the level of the free alcohols. So what does glucosylation do to the original molecule? Glycosylation of small molecules increases the stability of the molecules, which is shown here by the increase of the half-life of free uraniol when it is glucosylated. So it's only 28 days stable, whereas furanate glucoside shows a much higher stability. The water solubility is increased. Yeah, octyl glucoside is 20-fold more soluble or better soluble than octanol. I already showed you the compartmentation. So glucosylation is something like a flag for the cells to show where to transport these molecules. The bioavailability of the molecules, they change. Yeah, you can see here the concentration of quercetin in the plasma when you either administer quercetin 4' glucoside or quercetin 3 glutanoside. Yeah, so here much higher concentration of quercetin is present in the plasma, but when the 3 glutanoside is applied, the concentration stays longer in the plasma. The volatility decreases when the compound is glucosylated, and also the taste is different. Yeah, so naringenin is flavorless, whereas naringenin, the disaccharide, shows a bitter taste. So, and due to these properties, these glucosides are of interest for industry, for food industry, pharmaceutical industry, cosmetic industry, and also hygiene industry. Due to the stabilization, the molecules can be stored much longer. Due to the improved solubility, they can be applied in a matrix where the free alcohol maybe not be used. Yeah. Taste can be enhanced or modified. The drug can be targeted, and these molecules, in particular for the flavor industry, can be used as slow-release chemicals because there are enzymes which can specifically cleave the bond between the glycan, the sugar moiety, and the glycan, the volatile. Yeah? And when this bond is cleaved, yeah, the volatile is then released. So, and here you see during 1988 and 2015 a number of patents have been filed for the production and application of geraniol beta-glucoside, yeah, for industrial purposes, but until now the geraniol glucoside is too expensive and has not found use in any product. But there are some very interesting properties which have been shown by many publications. So here the function as slow release aroma chemicals, they can be applied, or the glucosides, aroma glucosides can be applied on human skin. Human skin is covered with the skin microbiome, and these microorganisms, they produce glucosidases, which are able to cleave the bond between the volatiles and the sugar molecule, and can release the flavor molecules, geraniol and citronellol. Yeah? If you have a look on your cosmetics, yeah, in about 50% of the lotions, the creams, and so on, yeah, geraniol is present. And as you can see here, especially the beta-glucosides are hydrolyzed. So the microbiome produces especially beta-glucosidases. Already after 2 hours, the volatiles can be detected, but the level is much higher after 6 hours. And here, when you analyze the different strains of the skin microbiome, yeah, they of course show different levels of beta-glucosidases and release different amounts of the volatiles. Yeah, so since humans have different skin microbiomes, they will probably release different amounts of these volatiles by their skin microorganisms. So, nice applications, but the compounds are too expensive. Why? Yeah, because there are, in principle, two pathways for the two procedures for the production. Chemical procedures and biochemical procedure. The chemical procedure relies still on the Königs-Knorr reaction. All what you can find is a modification of this basic reaction, yeah, involving heavy metals. Yeah, you have to work in water-free media, exclusion of light, yeah, and so. And the poor yield, so the product, of course, glucosides can be synthesized by the Königs-Knorr reaction, yeah, but yeah, due to the waste and due to the low yields, they are too expensive. Biochemical synthesis makes use of glucosidases, glucoside hydrolases, phosphorylases, and transglucosidases, which can be used to synthesize the glucosides, but in nature, these enzymes actually catalyze the reverse reaction, the hydrolysis of the glucosides. The real enzymes in nature which transfer a glucose residue to a glycone are the glucosyl transferases. Yeah? And although a number of glucosidases and glucosyl transferases have been used and been tested for the production of terpene glucosides and aroma glucosides, the costs of these glucosides is still too high for commercial applications. But during the last years, due to the sequencing of many plant genomes, more and more sequences of glucosyltransferases or plant glucosyltransferases are now available, and about 100 to 200 glucosyltransferases genes can be discovered in the different plant genomes. These enzymes, which all belong to the group 1 of glucosyltransferases out of 94 group in total, they catalyzed the transfer of a glucose moiety from UTP glucose to the glycan, to an acceptor, so the glucoside is produced. The structure of these proteins consists of 2 domains. One domain is responsible for the interaction with the acceptor, so it defines the specificity. The other domain is responsible for the interaction with the UTP sugar. This region contains also the plant secondary product glycosyltransferase motif. So this motif interacts with the UTP sugar, and there is also a backbone which holds the 2 domains in the correct distance, because the active site is located in the cleft between the 2 domains. And so, since the sequences are now available, there has been many research done on the specificity of these enzymes, and now we know that they are, fortunately for white biotechnology, are not very specific. Yeah? They can glucosylate a variety of molecules, yeah? And we tested different volatiles, and as you can see here, for instance, this gene or this enzyme from grape can glucosylate terpenoids, but can also glucosylate phenolic compounds, and also furanol, which is a major or the most important flavor molecule of strawberry. And so now we have tools available for the glucosylation of aroma chemicals, and since UTP glucose, which is a quite expensive cofactor, is necessary, we are performing the biosynthesis or the production by whole cell biocatalysts. So the E. coli cells which we are using are recycling UTP glucose, and so we grow, and other researchers normally grow the E. coli to high density. Then the production of the plant glucosyltransferase of the transgene is induced, and afterwards the acceptor is given to the E. coli cell, and fortunately the acceptors are taken up by the E. coli cells, and the products are then liberated to the media again. Yeah, so this is quite interesting. We don't know nothing about the transporters, yeah, but high levels of the glucosides can be extracted from the supernatant, and this was demonstrated for many different aroma compounds. Of course, there is still room for improvements, yeah, and one limitation is of course the pool of UTP glucose. There are different strategies, yeah. Either you can overexpress UTP glucose biosynthesis, or you can delete UTP consumption by competing pathways. Yeah, and we also recognize that there is a glucosidase activity by E. coli, which can also be reduced. So in order to increase the pool of UTP glucose, phosphoglucomutase has been increased, the expression of phosphoglucomutase has been increased, and UTP glucose pyrophosphorylase and this measure has increased the carbon flux to the glucoside by 8-fold. And here an example for the deletion of competing pathways. So UTP glucose is also consumed by UTP glucose dehydrogenase, by galactose 1-phosphate, urididyl transferase, and by UTP glucose for epimerase. And by reduction or by elimination of these enzymatic activities, a 2-fold, a 3-fold, and a 3-fold increase into the carbon flux to the glucoside could be realized. And only recently, a new method has been published for the recyclization of UTP glucose, which relies on sucrose synthase. This enzyme uses UTP and sucrose, the cheap sugar sucrose, and forms UTP glucose and fructose. So this enzyme sucrose synthase together with sucrose can be used in vitro assays to recycle UTP glucose from UTP, as can be seen here for the formation of curcumine diglucoside from curcumine in the presence of the sucrose synthase, which is also called SUSI. There is a tremendous increase in the concentration of the diglucoside when this recycling system is installed. And at the moment, we are working on sucrose, or we have transformed sucrose synthase into E. coli cells in order to perform this recyclization of UTP glucose in E. coli cell. And so far, the results are very promising. We obtain about 5 to 10 grams per liter of the terpene glucosides and also phenolic glucosides in the supernatant, and we believe that this system will be available soon to produce terpene glucosides and aroma glucosides in an economical way. So, summary, the glucosylation is one of the most important post-modification processes of small molecules and improves the physiochemical and biological properties, for example, of flavors and fragrances in terms of increased solubility, stability, and bioactivity. Enzyme-based glycosylation of small molecules is superior to chemical synthesis and has achieved significant progress due to advances in protein characterization and engineering, as well as exploitation of biosynthetic gene clusters of natural compounds. And in vitro and whole cell biocatalysts have been developed, which will allow the biotechnological production of small molecule glucosides on an industrial scale in the future. And we hope that next year we can start with a production. If you are interested, have a look on the homepage of 4Gene. Thank you for your attention. And